This is our second recording for Chapter 3. In our first recording, 3.1, we talked about the three components of the cell theory, some basic characteristics that all cells have in common, and then the two main categories of cell, prokaryotic and eukaryotic cells. In this recording, 3.2, we're going to talk through the cell organelles. Now notice this one is a little bit longer. We have in all about 12 slides to cover here, so just bear with me and let's try to talk through each one of these in turn. An organelle is simply a small structure, a membrane-bound structure typically, that carries out a specific function. Things like protein synthesis or processing and packaging and storage and communication. Here we have an animal cell. This is just a very stereotypical animal cell. They don't all look like this, of course, but you can see all the various organelles within and they're all labeled. Same thing with a plant cell over here. Now these are both eukaryotic cells. Remember, prokaryotes are bacteria, eukaryotes are everything else, and they contain all these various organelles. The first one we're going to take a look at are the cytoskeletal fibers. Cells have to have some sort of an internal structure just like your body does. You have a skeleton that holds you up. Well, a cell has this very complex network of microtubules and intermediate filaments and microfilaments that give it its shape and allow it to do the jobs that need to be done. So we have this network of crisscrossing proteins, and you can see in this beautiful image the cytoskeletal fibers have been highlighted in different colors. And you can see it's this complex web going all throughout the cell, and there's the nucleus in the middle. So we've got these three kinds. The microtubules, they're like train tracks. They allow for the movement of organelles and they assist in cell division. Intermediate fibers are more structural, they're more for strength, and then the microfilaments, they are also useful for movement, for moving things from one area of the cell to another. The cytoplasm, like I had said before, is the space between the nuclear membrane and the outer cell membrane. This jelly-like substance called cytosol fills the space. The space is the cytoplasm. Everything in there, the jelly substance as well as the organelles, are all called the cytosol. All right, the nucleus, the primary organelle within a eukaryotic cell, its main function is to store that DNA and keep it safe, keep it protected. It allows certain molecules to have access to the DNA when necessary. It's not enough just to maintain it in this organelle, but we have to be able to use it. So built within the membrane of the nucleus are these pores that allow certain molecules in and out. It's double membraned in structure, and within it there is a dense region where the ribosomal parts are going to be made. Now ribosomes are made from two different parts, the big part and the little part. Well, those two parts are going to be made right here in the nucleolus. It's not surrounded by its own membrane, it's just a dense region inside this larger nucleus. Next we have the endoplasmic reticulum, which is an extension of the nuclear membrane. It is a large system of folded membranes. You can see right here, it's just back and forth folds of membrane. There's two kinds. There's the rough and there's the smooth. They have different functions. The lumen is going to be the space within the membrane maze. So this is a great picture of the endoplasmic reticulum. It's these yellow bands. The space in here in the middle is called the lumen, and that's where proteins are going to be processed and packaged. Proteins and lipids are two of the products that come from inside this endoplasmic reticulum. Now the difference between the rough and the smooth is that the rough has these little ribosomes all over its surface, and you can see them really well here. They're the little red dots. These ribosomes are making proteins, and those proteins are going into the lumen where they're getting processed. The smooth endoplasmic reticulum has a couple different jobs, but it's basically the same type of a membrane structure. The ribosome is the site of protein synthesis. That's where proteins are made. So here's the same picture, but now we have the highlighted little red ribosomes. So these guys are pumping out proteins into the lumen of the endoplasmic reticulum. There are two different kinds. There are bound ribosomes. These are the ones that make proteins that need to be used either in the cell membrane itself or on the outside of the cell. So these little guys right here, they're all bound. They're stuck to the endoplasmic reticulum. Everything they make goes inside the lumen, and it stays contained and it stays safe there. The free ribosomes, like this guy right here, he's not bound to anything, and so he's free floating. The proteins that guy is going to make are the ones that are used inside the cell. So his protein construction is different than theirs. This guy's proteins are useful just out here in the cytosol of the cell. Next we have the Golgi apparatus. It looks like a stacked up layering of empty sacs. They're basically just membranes, and what happens is the endoplasmic reticulum is going to make those proteins, get them transferred into the lumen, and then send them off in little vacuoles, like these little guys right here, these little chambers. They're going to enter the Golgi apparatus and just kind of merge with this stack of membrane sacs and get processed and worked on a little bit, and then eventually get shipped out in other little bubbles and sent out to the cell membrane. So in this layering of stacks is where specific enzymes are going to process and work on the proteins before they finally get transported out. Mitochondria, very important organelle. You can see right here its shape is very bean-shaped. It kind of looks like a prokaryotic cell, but inside there's all these folded membranes. It has unique structures that allow enzymes or energy from food to be converted to usable chemical energy. Basically, it's the powerhouse of the cell. The mitochondria takes the energy that's stored in the food that you eat and puts it into a form that your cell can use. All these inner membranes, these inner folds, that's where that's taking place. They are believed to have at one point in time been independent prokaryotic cells that lived on their own, but have since moved inside of a cell and now live inside of eukaryotic cells. This is a symbiotic relationship. It's called endosymbiosis. Vacuoles. These are basically little storage chambers. All these little blue dots would be little vacuoles. They are fluid-filled sacs used for storage. Animal cells generally have lots and lots of small vacuoles, whereas a plant cell will have some small ones, but they generally have one very large central vacuole that's used for storage as well as support. So you can consider these something like the closets of the cell. The lysosome is a membrane-bound sac that contains digestive enzymes. When this guy engulfs something else, when a small vacuole moves inside, you can see these two have little bits of something inside of them. This lysosome has enzymes that can get to this little particle and break it down. It works as a defense. If a cell takes in a bacteria or a virus or something that needs to be destroyed, it encases it in a vacuole, and then that vacuole moves to the lysosome where the digestive enzymes then break down the little components and release them harmlessly out into the cell. Next we have the centrosome and the centrioles. These are a little less understood. They're not quite as easy to explain. The centrosome is a region. It's an area of the cytoplasm where the microtubules are going to be produced. So it's a space. The centrioles are the actual little organelle. And centrioles are made from some of those cytoskeletal fibers, the microtubules. Here you can see one centriole in a circle, and then here's the other centriole right beside it. They always appear in this configuration with perpendicular orientation to one another, and you can see it's made of little tubes. So here we're looking down on the top of it. Exactly what they do we're not really sure, but we know it's related to cell division and spindle fiber formation. Alright, cell wall and chloroplast. Now these two are special because they're only found in plant cells. Plant cells are still eukaryotes, but they have some different structures than animal cells do. One of them is a cell wall. Here you can see this is an Elodia leaf. This line is a cell wall going around one individual plant cell. The cell wall provides structure, it provides support, it gives the cell its shape and enables the cell to hold up the rest of the plant. The chloroplast is the other organelle that's found only in plant cells and some other specific protists or bacterial cells. The chloroplast is going to convert solar energy, which is just energy from the sun, that we can't use, it's going to convert that into usable energy, into the form of a sugar eventually. And we'll talk about this in a future chapter. But you can see this organelle has very specific structures inside of it, and these are important for converting that solar energy into usable chemical energy. All right, that is the end of 3.2. Next recording will be on 3.3.